My name's David Earhart, and I am a staff member here at the Carnegie Institution for Science on the Stanford campus.
So my name is Heather Meyer. I'm a postdoc in David Earhart's lab at the Carnegie Institute of Science, Department of Plant Biology, and I'm working on trying to understand how plants perceive seasonal temperatures to know when to flower.
Hi, I'm Stephen. I'm a postdoc in the Gittler lab here at Stanford, and I mostly work on neurodegenerative diseases, but I'm slowly starting to also work on plants to study stress responses.
Hi, my name is Cesar Cuevas Velázquez. I'm a postdoc in COSED in Anilab, and I am using intrinsically disordered proteins to develop fluorescence-based biosensors to track the environment within cells.
My name is Sue Ree. I'm a senior staff scientist at Carnegie Institution for Science in the Department of Plant Biology. My group works on a lot of different things. We're generally interested in how plants acquire traits to adapt to environmental conditions. And mostly we work on metabolic traits, but more recently we've been working on seed germination trait as well as organ size control.
Hi, my name is Benjamin Jin, and I am a research assistant within the RE lab at the Carnegie Institute. So what is liquid-liquid phase separation? We define this phenomenon as the ability for different substances which occupy the same volume to be able to both exhibit liquid-like behavior and yet also have the ability to disperse into different compartments, a process known as demixing. We can see this phenomenon most popularly when we create salad dressing, which we will demonstrate here using Chinese black vinegar and canola oil. Both of these substances, of course, exhibit clear liquid-like behavior, and yet, when we put all of them within the same volume, we know that they form completely separate compartments from one another. So why is this important? Well, it is important because life occurs within liquids. If you think about the cell and the cellular environment, specifically the cytoplasm, all of the reactions and the organization that happens within a cell occurs within liquids. Traditionally, this organization of the different reactions and processes that happens in cells have been ascribed to membrane-bound compartments, which we call organelles. However, recently scientists have discovered the existence of membrane-less compartments, which seem to have a myriad of different functions within the cell. These membrane-less compartments seem to remarkably exhibit liquid-like behavior even though they exist within the liquid environment of the cell, thereby producing the hypothesis that they must arise out of liquid-liquid phase separation. In studying this phenomenon, we will therefore be able to unravel a powerful biophysical process which seems to contribute to many of the processes that cells use to organize themselves.
First of all, I think there's a lot unknown about biophysical rules that govern how organisms and organs and cells behave in all of life sciences. And this phase separation seems to me like a very interesting way of thinking about how molecules and complexes might be working inside the cell and might provide a very interesting mechanical or biophysical explanation of function.
I think phase separation is super cool for maybe two reasons. First of all, phase separation gives us a novel biophysical means of how the cell achieves cellular subcompartmentalization, and this is important for studies of especially non-membrane bound organelles, so the nucleoli, stress granules, and so on. And secondly, phase separation is also giving us some insights in protein aggregation. So pretty unexpectedly, because it's a slightly different process, but for years we have known that this process is implicated in diseases, but also under conditions of stress and so on. And phase separation is now shining a new light on how this process happens and how the cell may actually prevent this process. So from even a therapeutic point of view, the study of phase separation will be crucial to understand how we can maybe one day treat these diseases associated with protein aggregation in the brain.
So I'm excited about the field of phase transition or phase separation, mostly because it's a new way of thinking about biology, and a lot of these proteins that are thought to phase separate might be involved in perception, and this is something in which we don't really understand a lot of how organisms perceive and sense their environment. So it sort of is opening up this new avenue of research.
Yeah, so I think that the phase separation field is very exciting because it provides a nice way to explain how cells compartmentalize themselves and how can they perform specific biochemistry in a controlled spatiotemporal manner.
So now we have this whole new world to explore, and I think we're seems like every few months a new report is coming up of a new compartment in a cell, and sometimes maybe now outside of cells, that are organized or created by these disordered proteins.
And finally, I think that there's a lot of potential to develop technology out of this biological process. So you can imagine that you can design synthetic compartments that are produced by a particular input and that perform a particular reaction that increase the yield of X or Y product that is desired for us.
I guess another reason I'm excited is that I think that, of course, I'm not, by far not the only person who's ever thought about this or discussed it, but this phase change seems to me to be like a really attractive idea in considering how life might have originated on the planet. Because one of the things we really need to explain to get life off the ground is how biological organization came about in the first place. And it seems that this phase change is something that you can get relatively easily. You can imagine how it could happen easier than some other kinds of organizational changes as early molecular evolution took place.
So I think this field has a lot of opportunities for plant biology in general, because most of the proteome of plants is not well understood still, and a lot of that is enriched with IDPs, or these intrinsically disordered proteins. And so I think that there's going to be a lot of cool biology that starts to come out in studying phase transition and intrinsically disordered proteins, and understanding broader mechanisms in plant development.
Yeah, so I think the field of phase separation in general, intrinsically disordered proteins in plant biology is not very well explored so far. So the field is very small. But I think that there's increasing interest on this type of process and how it plays a role in the biochemistry and the biology of plant cells.
I mentioned to you I study mostly neurodegenerative diseases, so why am I interested in plants? Well, phase separation is a universal mechanism. It happens across all living organisms, and especially it seems to be important in stress conditions. And we know that these stress conditions play a major role in these neurodegenerative diseases, but what we now want to do is study more how other cells, or non-human cells, have evolved mechanisms to cope with stress using phase separation. And plants are an ideal model organism to study stress responses, as they're continuously exposed to different temperatures, different salinity of the soil, and so on. And what mammals often do, like humans, we run away from stress. Plants can do that, so plants have really evolved intricate stress responses, which may now give us new clues of how we could use these type of processes as therapeutic approaches in neurodegenerative diseases.
And I think one thing we're really excited about in my group is the fact that phase change is very environmentally sensitive. And so in plant biology, we know that plants are not homeostatic in the same way that, say, mammals are. And we're really interested in how phase change may serve as a way of the organism to respond to the environment, to communicate with the environment. And we're interested, for example, in the possible role of phase change in temperature sensing, since phase change itself is very, very temperature sensitive. And these phase change compartments, they may have surprising roles in places we haven't looked very carefully yet. Like there are a number of proteins that are secreted, predicted to be secreted in plant cells that look highly disordered. And we're very curious about whether it's possible that disordered proteins could perform functions that we haven't discovered yet in parts of the cell that we don't study as heavily as, say, the nucleus itself.
We know very little about what goes on in plant cells, and so I think there's a lot more to discover. And some of the very recent findings in our lab indicate that proteins that might be involved in phase separation could play very interesting roles in regulating a lot of important processes ranging from germination to communication to controlling growth.